Good day. I am presenting our study on using wild Kappaphycus populations and selective breeding as sources of new cultivars for biomass production. This presentation is divided into two parts. The first part is on DNA barcoding and haplotyping of wild Kappaphycus, and the second part is on selective breeding. Kappaphycus is a primary source of raw material for Kappa carrageenan production with several applications in the food industry. It is also one of the sources of livelihood for coastal communities. Philippines is the first country that introduced seaweed farming in the early 1970s and is also the top seaweed producer until its production slowly declined in 2012. This decline in production has been associated to the clonal propagation of cultivars that reduces overall productivity and increased susceptibility to diseases and epiphytes as shown in these photos. And consequently, there is also a decrease in carrageenan yield and quality. Due to the low genetic diversity in the cultivars currently cultivated, there is a need to generate new strains to sustain Kappaphycus production and the carrageenan industry. Some of the techniques used to improve quality of available seed stacks include micropropagation and protoplast isolation and culture. Another way is to use wild populations with superior traits to generate new cultivars. In this regard, there is a need to assess wild Kappaphycus populations. To do this, we will use molecular markers for species identifications, which is the main objective of this study. We collected samples from the northern Philippines and central Philippines, that is in Bolinao, Pangasinan, and in Giwan, Eastern Samar. Species was first identified based on their morphology, and samples were preserved in ethanol for DNA barcoding. Molecular assays follow standard protocol from DNA extraction, PCR amplification, sequencing, and phylogenetic analysis, where multiple sequence alignment was performed and maximum likelihood tree was constructed. Samples from central Philippines in Samar was initially identified as Kappaphycus alvarezii based on gross morphology. Color ranges from green, brown, red, and shades in between, and some exhibited multicolored pigmentations. The phyli were erect with discoid or crystal holdfasts, and some holdfasts are attached to coralline substrate. Based on gross morphology, samples from northern Philippines was initially identified as Kappaphycus striatus. Phyli color ranges from shades of yellowish-green, yellowish-brown to dark brown. Habit is mostly prostrate or decumbent, with holdfasts that are inconspicuous haptera. Our initial identification was supported and confirmed by our molecular data, and the use of COX-2-3 marker was sufficient and effective in delineating between species. Moreover, we found two new haplotypes in Kappaphycus alvarezii, that is the CALB5 and CALB6, and the majority of the sequences are identical to the known haplotype previously reported by Dumilag et al. Our study suggests that these haplotypes are biogeographically localized in Giwan, Samar. For Kappaphycus striatus, we found new haplotype in northern Philippines on top of the commercially known haplotype 89. And this is the first report of wild Kappaphycus striatus in Pangasinan. Different individuals belonging to the same haplotype exhibited different morphologies, which is very interesting. For example, for CALB3 haplotype, as shown in this figure A, B, and C, They belong to the same haplotype, but they exhibit very distinct morphologies. On the other hand, Calb6, which is a new haplotype, exhibits very distinct morphology compared to Calb3. In Kappaphycus striatus, the differences in morphology is not as distinct as those observed in Kappaphycus alvarezii. In summary, our study confirmed the presence of wild Kappaphycus alvarezii in Samar and Kappaphycus striatus in Bolinao, Pangasinan. The newly generated barcodes increases reference materials in the databank of Kappaphycus, and the newly recognized haplotypes are potential alternatives for currently farmed strains, and we found diverse morphologies and phenotypes among wild Kappaphycus alvarezii and Kappaphycus striatus. For the future, there is the need to explore the unutilized reservoir of wild genotypes and to examine the potentials of the identified wild strains as alternative cultivars for crop improvement. And this could be done through examining the growth rates and carrageenan yield and quality of the identified cultivars and the resistance to epiphytes and diseases, and to do some field test planting. The use of more markers, as single or combined, is also needed to further assess genetic diversity, and the employment of genome-based assessment and comprehensive morpho-phenotype analysis is needed to elucidate the morphological variations among Kappaphycus species. Results of our study is now published in this paper. The second part of this presentation is on selective breeding as potential source of new cultivars. To start with, I would like to introduce the triphasic life history of eucheumatoids. You can see here the haploid male and female gametophyte, which undergo fertilization and produces the carposporophyte that is carried by the haploid female gametophyte in the cystocarp. The carposporophyte produces carpospores that germinates into tetrasporophyte, and the tetrasporophyte produces tetraspores that germinates into male and female gametophyte. This is a sample tetrasporophytic plant, which could be distinguished by this tetrasporangal nematecia, which is very distinct here, the whitish translucent structure. And this is the male and female gametophytes. You could see here the male gametophyte with this protruding spermatangial nematecia, and you have here the female gametophytes wherein it has a wart-like structure, which is basically is now the carposporophyte. And here is we're showing spore release and germination. So this is basically carpospores released from the carposporophyte, and this is the tetraspores released from the tetrasporophyte, and this is the germinating spores attached on a glass slide. This is just a quick rundown of the natural selective breeding that we are doing in the lab. So here on the left-hand side column, you will see the parental generation of the tetrasporophyte and a female gametophyte with the cystocarp. So we induce spore release as shown here in this picture, and the released spores is basically cultivated in the lab until they grow into sizable germlings, and this is brought into the land-based tank cultivation system, and this is basically now the F1 generation of the next generation of gametophytes and tetrasporophytes. For our very recent experiment, we basically collected 6 mother plants for spore shedding experiment. And these are the carposporophyte with different morphologies and color, and the tetrasporophytes here, three mother plant tetrasporophyte. Same thing was done, spore release and germination was conducted in the laboratory, and after a few months they basically germinated into the F1 generation. So these are basically different age and size class of the next generation tetrasporophytes, and this is the different size class and age class of the next generation gametophytes. And as they grow older, you could see here the gametophytes. They exhibit distinct morphologies. You have the male and the female gametophytes, and also, very interestingly, you could also observe the different color expression. In the subsequent slide presentation, I will be showing the mother plant and the progenies. For example, here, this is the mother plant KACRC, and this is the progeny of this mother plant, and you could see diverse expression of color and morphology. And in this case, this is the mother plant KACRR, and you could also see here the progenies exhibiting different color and branching patterns. Same as with the KACRS. So you could see here the distinct coloration. So we suspect that there might be some genetic basis in color expression, because all of these siblings are cultivated on the same environmental condition in our hatchery cultivation system. And this is now the progenies of the parental tetrasporophyte. So you could see here the 8-month-old gametophyte and the male and female gametophyte. So different branching patterns, and you could also see here a female gametophyte with this cystocarps. And this is the progenies of the mother plant KATRO. You could see different branching patterns and different color expression, and we suspect that there is a chimera here, no? Where in one plant we observe a female branch and a male branch. In another male plant we see a cystocarp, no? So very interesting. And this is the progenies of KATRQ. This is the mother plant, and this is the different F1 generation. So these are basically siblings, and you could see here multicolor female gametophyte and a very a nice red colored male gametophyte and female gametophyte. So we are now monitoring their growth and how robust they are. So in terms of their susceptibility to endophytes and epiphytes and diseases, and we observe that the F1 generations, the different siblings have different susceptibility to epiphytism and to diseases. And we also observe evidence of recovery from disease. So these are some representations of the epiphytes and disease that we observe. So you could see here epiphytes on the surface, and here also. And this is basically a disease that there is some kind of mucilage on the surface that it is eating away the tissue. And here we observe evidence of wound healing in deceased plants. So you could see here, this is a thin film of milky white mucilage. And then when as they grow further, you could observe the healing of the wound here. Okay, so this is basically a scar of the previously diseased tissue. And we are also separately drying biomass from different siblings or from different cultivars, and they will be they will be analyzed for carrageenan yield and quality. This is a sample of our setup in Bulinaw, Pangasinan. So we have more than 100 aquaria. So each of these aquarium is basically holding different strains, different different cultivars, eventually different cultivars from different parental plants. And this is our raceway setup. So this is Kappaphycus striatus from the wild, and we are maintaining them in this raceway, and they are growing very nicely. And this is our other raceway setup, and yeah, this is showing the rest of our cultivars being cultivated in aquarium and raceway. And this is a closer look on the different cultivars. So these are all Kappaphycus. This is Kappaphycus striatus. This is Kappaphycus alvarezii. So cuttings. Now we are now clonally propagating them. So they exhibit different colors. So these are basically different strains of Kappaphycus alvarezii. You could see they have different branching patterns, different colors, very distinct morphology. So this is a commercially known haplotype of Kappaphycus that we also cultivate. We are trying to compare the growth rate of the commercially known haplotypes, compare it with the growth rate of our new haplotypes or our new strains. That ends my talk, and I would like to thank the research staff involved in this study and all the funding agencies that supported our research. Thank you very much for listening.